Welcome to the European College of Neuropsychopharmacology podcast, where we interview the most interesting people in translational neuroscience.
Hello, welcome to a new episode of the ECMP podcast. My name is Mairéad and I am a final year PhD student in University College Dublin. And today I'm going to be speaking with the researcher, Dr. Simone Macrì.
First of all, thanks for this invitation, which I like a lot. I'm really happy to be here and share my views as to certain aspects and mostly talking about the research I'm doing at the moment. As you just said, I'm Simone Macrì, and by training and the definition I like most is a comparative psychologist. So I have relatively hybrid education, how shall I say. So I studied experimental psychology in Rome, then I did my PhD in behavioral neuroscience at the ETH in Zurich, and then I had some experience in the US at McLean's Hospital and at New York University. And currently I'm working in Italy at the Istituto Superiore di Sanità, which is the Italian version of the NIH, essentially.
Well, very broad background, and you've been to lots of different places. A little bit more than probably with me in that I suppose you would call me an early career researcher, is the term that they would use. I am in my final year of my PhD, very much focused in Dublin. My background is in genetics. I did my master's in neuroscience and then continued on in Dublin with my PhD. Largely, I was intending on traveling a little more, but COVID put a stop to that. So I started out my PhD in 2020. But really interesting to hear about you and all of your background and all the different places that you've been.
Well, first of all, fortunately enough, you have time to travel after your PhD. So
finally.
You don't have to worry too much about it. Because this is, I guess, one of the nicest parts of our job, that it gives us the possibility to travel a lot. And as I said, I traveled a bit, not that much, but in principle, I'm Italian. Mm-hmm. And I've spent something like 5 years of my life outside Italy, which was very good for me. All different places gave me a lot. And a lot of different things, different perspectives. And I've tried, not sure I've managed, but I've tried to grasp as much as possible from each single place I've been. Zurich was fantastic for me because it taught me how to think about science in terms of projects, in terms of taking one idea from the idea itself to a way to operationalize this idea into experiments and execute an entire project to try and answer some questions. And at the end, sometimes you end up with an answer that opens up other questions. So I like this a lot. And in Zurich, I studied the neonatal programming of the stress system. So what happens in your neonatal environment, how that might have enduring effects on how you will behave once adult. And behavior has been my key topic. And something I've done, as I said, I like to see myself as a comparative psychologist. And in that respect, I've studied different species, mostly rodents, mice and rats. But I had the opportunity to work with zebrafish in New York. Once back in Rome, I'm also having a chance to have some projects in which human participants, human volunteers are involved. So yeah, that's for what concerns Zurich. Then in Boston, I've tried kind of at a project that was very close to what I did in Zurich. Then a completely different topic with zebrafish work in New York. But now what I'm mostly interested in is experimental models of human psychiatric disorders. Mm-hmm. I started off long ago with depression, but now I'm getting more and more interested into cognitive functions, specifically executive functions. And very recently, well, as if 4 years can be considered very recently, I started focusing more on the interaction between metabolism and behavior. That's the key interest at the moment.
Yeah. And I think that brings us really nicely into the idea of the PRIME consortium. So to give a little bit of an introduction into what PRIME is, it's a European consortium. It started out in 2020. And the focus of PRIME, which stands for the Prevention and Remediation of Insulin Multimorbidity in Europe, is looking into the role of insulin, so the crucial hormone involved in that glucose regulation, and what it does in terms of its role in neuropsychiatric disorders. So we're looking at disorders including obsessive compulsive disorder, autism, and various symptoms that go along with that, and looking at the role that insulin may play. So the consortium has multiple different groups and work packages. We have groups more so focused on the human clinical side. We have the genetics people, and then we have those of us looking more at the animal work. And then that's where you come in, Simona, with your experimental models that you talked about briefly. So what is your contribution to PRIME? How have you fit into that consortium?
So let me start saying something. I could have not introduced it in a better way. You've just
Great.
done a wonderful introduction. Yes, and as you just said, I had a lucky opportunity to coordinate the animal model work package. And what we're doing in that experimental model is to try and model or mimic what you just said on a human scale into experimental models. So essentially, we're all interested in, you've just introduced the role of insulin. Most of us are somewhat familiar with the role of insulin in diabetes. And what we want to do is to extend the understanding of the role of insulin at the brain level. So insulin does not just affect your metabolism, your peripheral metabolism, it also affects your brain. Mm-hmm. That's the key premise of PRIME. And we are trying to leverage experimental models to bring something new, something additional at the mechanistic level. So we're trying to investigate the mechanisms of how insulin in the brain and how insulin in the periphery also affect your behavior. So we know, for example, and there's ample literature outside, saying that Alzheimer's disease often co-occurs with diabetes. And likewise, we know that many diabetic patients also have cognitive symptoms that might end up with behavioral rigidity, for example, but also milder forms of cognitive impairments. And what we're doing is trying and translate this information backwards to experimental models to then be in the position to identify something, especially in terms of mechanisms, that can be back translated to humans, hopefully in, I would say, in the mid to long term, into novel therapies, trying and tackling both somatic and mental disturbances. And for this very purpose, we are leveraging experimental models that resemble type 2 diabetes quite well. They are mice that are called tally-ho mice. And I've learned from British friends that tally-ho is something that you use when you start the fox hunt or something like that. That's the story I've been told.
Yeah, I think the word tally-ho has been used for back, I think, in medieval times in Britain, when you threw things, waste out your window onto the street below, they would yell tally-ho.
Okay.
And then the people down below would know to take cover because anything could be falling down from above you. So that's where the word came from, I think, at least one of the origins, and then also the hunting, as you mentioned as well.
Yeah, well, why that has been given to some mice that have some close similarity, some close proximity to type 2 diabetes, I have no idea. It wasn't me to give this name. But anyhow, what these mice look like is that they have hyperglycemia, they have, which is a core symptom of type 2 diabetes. They have insulin resistance, they have alterations in the Langerhans islets and all the mechanisms that are related to insulin. And they've been used extensively to investigate type 2 diabetes. But recently, with all the contribution of the colleagues of the work package animal models in PRIME, that are David Slattery, Aito Leary, yourself, Jeffrey Glennon, and some other students from our lab, Edoardo Pisa, Martina Presta, Angela Ottomana. I really want to name them all because it's a collaborative effort. All of us identified some behavioral alterations in these mice that closely resemble some of the alterations typical of some psychiatric conditions. They have behavioral inflexibility. So if they are required, if they're asked to go left and right, either they always go left or they always go right. This is a very simple example of how their behavior tends to be relatively inflexible. We took this information to the next level, identifying alterations in executive functions. Executive functions are those type of functions that essentially allow us to keep track of what's happening and what we should do next in order to accomplish a certain goal. I mean, this is, to say it in very simple terms, how you translate this into mice. It's a difficult story, but it's feasible. It's very feasible. I mean, in psychology, there's a task that is called the Wisconsin car sorting task that addresses this very function. And we took that task and translated it to mice. And what we do is we train mice to seek for a specific odor, for example, to get a reward. And then we make the task a little more complex. So they have to identify this odor, but which is confounded by many other factors, but still they have to follow that odor to get their own reward. And the first thing I've told you, it's called the simple discrimination. So go towards cinnamon instead of sage. Follow a given odor. And then this odor is confounded by the digging medium in which you have to dig, get your reward. So this is a very species, mouse relevant task. And this is called the compound discrimination. So they still have to ignore irrelevant dimensions. Once they've done that, we make another trick, so to speak. And what we do is to do the reversal of this discrimination. So essentially, no longer cinnamon, but sage now. And then we do an intradimensional shift. So we change the orders, but still they know that the relevant dimension is odor. And then even the extra-dimensional shift. They no longer have to pay attention to orders, but to the digging medium.
Okay.
And so, and when I said that they have to pay attention to, it's because this task is considered to be highly relevant for attention and for executive functions and for behavioral rigidity. Because we see that these tally-ho mice, for example, do not switch as easily as controls. But what was our next step? Once we did what in comparative psychology is called phase validity. So identify a similarity of symptoms between humans and experimental models. We tested the pharmacological validity. So what we did was to give these mice metformin, which is the go-to drug, one of the go-to drugs when it comes to treating type 2 diabetes. And we've observed that metformin is capable of reducing the baseline hyperglycemia. So it does what we expect it to do in diabetes, but also to compensate for the deficits. So that means that this model now is apt to novel investigations. And next to this tally-ho model, we also have additional experimental models, one being the one you're working with. So you may want to tell us something about it. It's the called KCNQ1 experimental model. And then I'll tell you the third one, but why don't you say something about that?
Yeah, I mean, it's very interesting what you're talking about with the tally-ho models and all of the different interventions there, because it's a pretty new area. Well, not new, but the idea of the brain being connected with the body and insulin having such an impact in brain as well, and not just peripherally, is something that, is really opening up in the neuroscience world, I think. The other model that you were referring to is the KCNQ1 model, and that's what we're looking at as well in Prime. So over in Dublin at the moment, we have this KCNQ1 knockout. So KCNQ1 encodes a voltage-gated potassium channel. It's expressed in many different tissues across the body, brain, also in thyroid and pancreas, all of these different tissue types. And essentially what we feel is that this may be having an impact on behaviors through various different mechanisms, and also an impact on insulin regulation and insulin metabolism as well. What we were doing in Dublin for the last year or so really is just characterizing the behaviors that these mice have. So looking at these KCNQ1 mice, looking at is there changes in their anxiety, is there changes in their compulsivity, and then some of the behaviors that you mentioned as well in terms of their selective attention to particular aspects like digging mediums, rewards to odors like sage, cinnamon, all those kinds of things, and seeing are they capable of learning, can they switch their behaviors, are they making mistakes like the controls. And that's something that we're looking at in the KCNQ1 models as well. In addition to also having a bit of an investigation, I suppose you could say, into the mechanisms underlying the behavioral changes in your tally hose as well. So we've done a lot of proteomic analysis, looking at different brain regions, blood as well, and just trying to run analyses to see what are the common themes. Is there anything coming up that we can identify as potentially a druggable mechanism in the future, which is another side. There's the flip side to the behavior that you're doing, the tissue work in terms of looking at those mechanisms and what could be happening as well behind the scenes.
Yeah, again, you've said things right on the topic, spot on, so to say, because I guess what it shows is the level of complementarity among the different sides that are working together in Prime. And what we've done was to start from the phenotype, and what you are unraveling are I would call them the endophenotypes. And to add to this level of complementarity, we said at the beginning that also Alzheimer's disease is frequently associated with type 2 diabetes. And what the third component of our intermingling work package is the Goethe University in Frankfurt, where David Slattery and Ayat O'Leary are, and they're working on an experimental model of Alzheimer's disease. Mm-hmm. So in Rome, we started from an experimental model of type 2 diabetes, attempted to identify cognitive abnormalities. In Germany, they're starting from Alzheimer's to identify metabolic disturbances and the associated mechanisms. And what you're doing is to go even deeper into the mechanisms. And I guess this is one of the things I like most about Prime, the fact that the different sites, different groups, different work packages are all mingling with one another. There's a cross-disciplinarity, which I like a lot. Because as you said at the very beginning, in the introduction, it's not just animal work. It's animal work plus epidemiology, plus genetics, plus cohort studies, plus clinical investigation. So this is something I like a lot. And it also, from my perspective, it also moves forward to the goals, to the achievable goals of Prime. What I see are some shorter-term goals that can have an impact on general public, patient populations, and that is kind of that can happen quite soon. And here I refer mainly to the work done also in Germany by Silke Matura, which I guess was part of another episode of this podcast. And I would encourage everyone to listen to it too. And they're using this mobile health technology and ecological momentary assessments to somehow first describe the behavior of patients and also to provide some insights as to which behaviors are preferable to maintain your glucose concentrations in a normal range, and now how that can benefit your cognitive capabilities. And next to it, there's the Predimed Plus study, which is done in Spain. And what they're doing there is to test the role of the Mediterranean diet on the metabolic and cognitive associations, so to speak. So there is a lot. And I get that this information can be very soon translated into effective approaches, not only for therapy, but also for prevention. So the more we access the public population, the higher the impact can be. And this, I guess, it's something that can be done on a relatively short span. And what will take longer, and this is one of the main hurdles or challenges I am personally facing. So how much will it take from what I'm doing to go from the lab into society? That might take longer, but that identifying common mechanisms, identifying drugs that were originally purposed for a given disturbance and demonstrate that they can also work on other phenotypes. So something given for type 2 diabetes, we are preliminary showing that it also has benefits on cognitive abnormalities. That means that in the long term, it's not something that I would encourage people doing now, not at all. But in the long term, that might help repurpose some drugs that are already there, that are already available, and that have already been demonstrated to be safe. Identifying the mechanisms, it's even a bigger leap. That probably means that new druggable targets can be designed. This complementarity, this sort of fractal structure of the project is something that I guess constitutes one of the primary key aspects of the project itself.
No, I think it's interesting, the idea that you mentioned about almost the repurposing of already available medications and drugs, rather than starting from the drawing board and trying to design something completely new. We can say, look, this is the mechanism that we think is important. These drugs already exist. Let's have a look and see what they can do. And that's really efficient, I suppose.
Yeah, it is. And this brings back to another thing you said before, like the so-called novelty of the association between metabolic disturbances and mental disturbances. And you said, yeah, it's nice to look backwards and see what's already out there. And I would say that the vestigial prime, so the ancient prime, is a paper from 1919, so 104 years ago. And it was from Coy, which identified alterations in glucose, increases in glucose in many psychiatric patients. His interpretation was that actually the psychiatric disturbances resulted in the glucose abnormalities and not the other way around. So the idea was that the behaviors associated with the altered brain pathways, so to speak, were the cause of alterations in glucose metabolism.
Okay.
And he did not talk about insulin in that seminal paper simply because insulin hadn't been discovered yet. I guess insulin had been discovered a couple of years after the paper has been published. And I guess this is something that I like a lot. Sometimes I like going back to very, very old literature and see that there are some aspects that are already there and that we should try to bring them back to life. Another, talking about my history as a scientist, life history, so to speak, is one of the most influential papers I've ever read. It's something that has been written in 1949 by Frank Beach, and it was essentially making fun of comparative psychologists, being himself a comparative psychologist. And he said that back then, most of the research was done in laboratory albino rats. And he said that essentially we're looking for the hidden mechanisms of human behavior. But what we're coming up with is the behavior of laboratory albino rats. And he said we really should go backwards to the 1900s, so of the previous century.
Yeah.
And he said that we used to do things in 9 different species before making a leap to humans long ago. Now we only work on laboratory rats.
Yeah.
So that's why I like the idea of working with different species to try and identify common mechanisms.
Yeah, like the multidisciplinary approach is important in that sense. And that was something else I was going to ask you about, actually, is that your work is very much focused on the animal side of looking into the neuropsychiatric disorders, so what behaviors are altered in animals. To the listeners out there who might be thinking in their mind, oh, whatever, it's only a mouse, how accurate can it really be? What would you say to that? Do you think that mice have value in representing these disorders, or do you think there's more that can be done?
I would say yes. So it's a sort of vague answer to it. Because the reason is that, first of all, I would tell them, yes, mice and experimental models have a huge value, tremendous value. This is undisputable. We know from Darwin that we share common mechanisms. We know mice, for example, they have to behave. They have their own goals. They have to pay attention to stimuli. They have to remember. they have to do lots and lots of things that we as humans have to do too. Likewise, as behavioral scientists or as comparative psychologists, we should be very careful. We should not go out and make big claims. We cannot say, I've identified the pill of a given domain. I've identified the mechanism of a given domain. We have to be very careful. We have to be very careful when we use or devise novel experimental tasks. One thing I like a lot about the attentional set shifting task I've mentioned earlier is the fact that it puts the mouse, the rodent, on top. So all the stimuli are devised to be species relevant. So what they have to do to get their food is to dig into something. And mice are great at that. They dig to build their own nests. They have to use others to identify where food is. And they're fantastic with their senses when it comes to with their olfaction. They're fantastic. They're perfect with that. I've also mentioned the digging medium, and with their vibrisses, with the whiskers, they're very great at perceiving where they're digging, the texture of where they're digging. So it's a species-relevant task. And I can like it so much because I did not develop it at all. I wouldn't brag at all about it. But it's been originally the first paper that I remember is from Verity Brown. And they've done that in rats. And then Giovanni Colacicco, Joe Gardner, and Anno Wurbel this translated to mice. That's why I'm saying that we have to be very careful. We really need to make sure that what we are saying is exactly what we've done, that we have a precise way to interpret certain behaviors. We don't have to come out loud with something that can be interpreted in different ways. So I guess that this is also something that I see as a challenge, is how do we translate this information from a laboratory to the public. So yes, experimental models are necessary. We cannot live without them yet, but the way in which I read things at times seem to be at best overinterpretation, and we call it like that.
Yeah.
So that is one of the key challenges I see in the field.
Just about being careful about not overinflating your findings.
Yes.
Yeah. At the moment in Dublin, we're also working. So we're working with mice, like what your lab is doing over in Italy. We also have recently started working with flies, or with Drosophila, which takes that idea of not overinflating your results to a whole new level, because we're looking at behavior. With mice, you can probably make certain claims about whether they, maybe feel anxiety or things like that. With flies, you have to be very, very careful. Again, it's really just about understanding the model and knowing exactly what every reading means, and not, going too far in terms of your interpretations, particularly with very small models like that. Useful for things like genetic manipulations, because they're very easy to breed and they're very easy to manipulate. The same with C. elegans or worms as well. That's very easy in that sense, but it's just being very, very careful, particularly when it comes to behavior, and not saying something that isn't necessarily the case.
Well, I couldn't agree more on that. As I said, I've also tried to work. I'm also working with species that are simpler than mice, like zebrafish. But one thing which was, not a major contribution to science, but at some point with my colleague in New York, Maurizio Porfiri, we've realized that most of zebrafish behavioral work has been, first of all, back translated from mice to zebrafish, which was fair, and also the tools you use to evaluate it have been translated from mice to zebrafish. The tongues are more or less the same size of the apparatuses we use for mice, and then you would put a camera on top or a camera on front of the tongue to study them. Well, although that seemed rather surprising to us, there weren't many papers that kept something into account, which is the fact that fish do not swim in 2 dimensions. They swim in 3 dimensions. That means one camera is not sufficient. You need 2 of them. You need to get an entire 3D motion and there we've shown, using some very complex ways of doing it, that some studies originally conducted in zebrafish probably led to inappropriate conclusions, simply because looking at them in 2 dimensions rather than 3
You miss a lot of information.
You missed a lot of information.
Yeah.
As I said, yes, experimental models are necessary. By the same token, we should be very careful and we should always question ourselves. Is this the only interpretation I can give to these results? Did I get the results the way they should have been collected?
Yeah.
So all these type of things need to be taken care of very, very carefully.
And then I suppose to zoom out a little bit from Prime and from all of those different contributions, more about you, and then could you tell us a little bit more about why you picked your field of research, why you went into this area at all?
Yeah. Can I say that I was not tall enough to become a basketball player? No, I don't think that's... Actually, I was not even fast enough, and I didn't have the required skills either. So, yeah, but seriously speaking, originally I was very, very much interested in human behavior. So that has always been my interest, and primarily behavior. That's the reason why I studied psychology, and the reason why I drifted from human psychology to comparative psychology is because back then I had the probably wrong impression that in the lab, you could control variables much more easily than what you could do with humans. And I really felt like I wanted to have a controllable system. I ended up realizing that it's not that easy to control confounding factors in rodents, and that human psychologists do a fantastic job in controlling confounding factors. So it's like I started off with 2 misconceptions, with 1 key goal and 2 misconceptions.
Yeah, because you live and learn.
No, it's a continuing learning process. That is something I can do. I can assure you.
Yeah.
And how do you foresee your future?
My future, I think, I'm coming now into the final year of my PhD. I think I would like to do a postdoc. It's just figuring out which aspects of my research I'd like to take further, I think, at this point. So there were a few different findings that we did discover through the work with your tally hose and then also with my own KCNQ1 mice that were very much of interest to me, including the mechanisms behind this compulsive behavior, and then also the idea of sex differences as well, which was something that I noted through a lot of my analyses over the last couple of years. So it's really just thinking about which aspects I'd like to take further and finding that lab that would be able to support that. Then there's obviously the ever-present question of funding and money and where I'm going to find that, and then just where to go as well. So it's an exciting time in terms of planning ahead and seeing where I'm going to go. I am from Ireland, from Dublin. Anyone who knows Dublin will know that the rent is absolutely crazy at the moment. So it's just trying to find somewhere with a more livable expense, like cost of living, I think as well, is another aspect that I'd be considering. Unfortunately, that has to come into play. Difficult for the, I suppose, early career researchers out there might also have that same issue as well. It's really just by finding, what I'm interested in, and then just where is the easiest and most logical place to take the next step.
Yeah, but you said several things that are really, really crucial, that you don't just have to take into account your scientific goals, but that you really have to take care of your work-life balance. That is something that we really need to be mindful about. This I'm sure of. I mean, some people can manage easily having a work-life, an appropriate and adequate work-life balance. Some others don't. I don't have the numbers here with me. I can just talk about anecdotal experience, and the experience has been extremely, the range of experiences I've seen is extremely high. So I've seen people that managed very well to keep their own lab going, etc. Some others overly biased their lives towards the lab. And I mean, that is also wrong what I just said. From my perspective, they skewed too much their lives toward one or the other side, and there are not just two sides. There's many things in life, but perhaps they were very happy with that too. So it's like, this is really personal perspective, and I guess everyone should be mindful of what they really have as the core aspects of their lives and follow all of them. So it's always good to follow advice from more established people. By the same token, they're not you.
Yeah.
They're else than you. So take them into account, consider them carefully, but don't worry about doing something that some established scientists would advise you to.
Yeah, it's very personal, I guess, everyone's decision.
Yeah.
The other aspect as well is one thing I found from working in Prime and then having a lot of international collaborators is sometimes there is a cultural difference to work. Irish people were very chill. We have a lot of amazing scientists, but I think we maybe have that slightly more relaxed attitude compared to maybe, I mean, as a generalization, obviously, compared to maybe some other places which would have maybe be more strict on deadlines. Obviously, everyone is different. There's something to be aware of as a general question in terms of where I'm going as well in the future.
Yeah, I mean, I don't know. I mean, granted, I've seen that going differently among people and labs. I wouldn't necessarily put a tag on a given
Everyone is the exact same. No.
a given nationality, so to speak. But there are some people that are, and I'm one of those. I mean, I tend to freak out if I'm close to a deadline.
Yeah.
And I feel like I'm not prepared for it. Some others are much more relaxed. And again, you see successful scientists that seem overly relaxed, but then they manage to accomplish everything they need to accomplish. Some others that behave as if they were absolutely strict about deadlines and miss them. You can make mistakes.
Yeah.
So if you miss a deadline, it's not the end of the world. I mean, I take it very badly. If I miss a deadline, I take it very bad. Yet, that's one of the key aspects that, according to me, especially early career scientists have to familiarize with, that success happens hand in hand with the insuccess. So sometimes it can be 90% success, 10% insuccess. Some other times it can be 50-50 or 10-90. I mean, that is something that, according to me, needs to be learned very early on, which is sometimes you fail. Fine. I mean, keep doing what you're doing. And especially when it comes to science, most instances, we have a committee evaluating us, referees. It's individuals. And individuals can like what you are doing or not. This does not say anything about you. And personal perspectives are very welcome. And I entirely trust those who select referees, committees, etc., to give a view that is an informed view, honest view, and some people may like what we're doing or not. Well, once we find people don't like us in terms of what we're doing, and some other times we do find people that like it.
Once you enjoy what you're doing, don't take the criticism too personally and just move on.
Don't take it personally, like Michael Jordan series, the Last Dance, and then I took it personally. That happens to me. Sometimes it happened. I mean, I'm not that nice.
You just have that quote on your wall and you remind yourself every time.
The background on Teams. It's Michael Jordan. I took it personally.
Okay, so I suppose one last question to finish off is you had mentioned to me before that you did some very unusual work at one stage with the Italian Space Agency.
Yeah.
Would you like to talk to us a little bit about that?
Oh, yeah, I'm particularly proud about it too. So at some point, so I got involved by some people at the Italian Space Agency. What they're doing, and this with all the international space agency, like the European ones and the NASA, etc., they are trying to plan the first manned mission to Mars. So when there will be a crew flying to Mars. And there is one problem that they need to solve among a million other problems, which is what will they eat? And taking on board weight in terms of what they have to eat, it's extremely costly. It's unbelievably costly. So one thing they're trying to develop also to promote the well-being of the crew is to devise these small gardens, so to speak, these vegetable gardens that are called BLSS, which stands for Bioregenerative Life Support Systems. So they build up small boxes in which they can have gardens, essentially. And this has nothing to do with me, of course.
Your gardening skills are
very pathetic. But then what they involved me for is that they identified some species that have a very good potential to be grown on board. And then I said, can you help us identify something that might promote individual well-being, that might promote cognitive capabilities, or at least contrast the effect of prolonged isolation and chronic isolation, which is this 1.5 years flight to Mars? Can you prevent a sort of short-term mild cognitive decline associated with the stress that astronauts will perceive? And then we came out with these cyclory roots that are used a lot to extract inulin. Inulin is a polysaccharide that has been suggested to have it's a prebiotic that has been suggested to have pro-cognitive capabilities. And so we had someone growing these cyclory roots into a BLSS on Earth, not in the space yet. And then we took this and gave it to mice exposed to a prolonged chronic stress model. And actually we've seen that chronic stress, and that was quite predictable, affected executive functions. But then a prolonged exposure to a diet rich in inulin in these cyclory roots essentially contrasted the detrimental effects. So as I said, I like having sort of open gaze as to what I can do.
I mean, yeah, that's very cool, though, to use your behavioral animal work into something entirely separate, working with the space agency. So will you be then growing some of these plants yourself, or? Well,
honestly, not myself, but my dad is doing. He has a small vegetable garden, and is growing it now. Well, actually, I should get the cyclory roots soon. I'll let you know. The leaves are used a lot as food, and the roots are not used that much. Yet, in one part of Italy, which is Liguria, they eat these roots a lot. They are kind of palatable. They're somewhat sour.
Okay.
So you really have to...
You have to kind of persist with it.
Well, not that much. I mean, you cannot do it that much while flying to Mars. But in principle, yes. I mean, they won't have chances to cook that much.
Yeah.
But recently, we applied for a small grant to actually bring processed cyclory roots on the International Space Station. I also like it a lot. I mean, the fact that you can translate something and using a food rather than a drug, that you can offer it to astronauts and they can say, I like it or not. I mean, it's something that is already present on our tables, so to speak. And I like it a lot.
Again, it's the idea of repurposing something that already exists for your purpose here.
Yes.
Great. Well, we'll wrap it up here, but thank you so much for chatting with us.
Well, thank you very much for the invitation. Really, really glad.
Thanks for listening to this podcast from the European College of Neuropsychopharmacology. The views and opinions expressed in this podcast are those of the interviewee and not necessarily the official position of the ECNP. And of course, this should not be the basis for clinical treatment decisions.